moving now into the area of mood disorders, our recipient here of the Colvin Prize, which was endowed by the Colvin family, is a giant in our field and a good friend for many, many years, and is also international. So far, all of our awardees have been born outside the United States and have been educated, for the most part, outside the United States. So the recipient of the Colvin Prize for mood disorders this year is J. John Mann, who is the Vice Chair for Research at New York State Psychiatric Institute in Columbia, just up the road. He was from Melbourne, Australia originally, was educated there, came to the United States, and actually spent quite a bit of time in New York trying to get a job at various universities. He was in almost every major medical center in the city of New York and had faculty appointments at NYU, at Cornell, and at Rockefeller before going to Pittsburgh, where he was recruited by the legendary Tom Detry there for many, many years before being invited to come to New York State Psychiatric Institute, where he is the Paul Jensen Professor, Vice Chair for Research, Scientific Director of a PET Center, and many, many, many other positions of leadership. He, as I mentioned, is a giant in our field, has made major contributions to our understanding of mood disorders, schizophrenia in particular, suicide in particular. He's made major contributions to brain imaging and genetics, and today has got a very broad and interesting topic to present entitled Unlocking the Mysteries of Mood Disorders by Science Instead of by Guesswork. Dr. Mann? Congratulations.
Mm-hmm. Thanks, Bob. Good morning. It's a tremendous honor to receive this award. And I'm deeply appreciative to the foundation, BBRF, and obviously to the committee that selected me for this. I'm going to talk a little bit about some of the research that I have done together with a group of highly capable, talented colleagues. As other people have said, it takes a village to do this kind of research. I'm lucky enough to be the spokesman. Of course, this work is funded by NIMH and the BBRF. I do get royalties for the use of the Columbia Suicide Severity Rating Scale, but it's free for use anywhere by any researcher, any not-for-profit organization or company. I was attracted to psychiatry originally. I was in the middle of doing my internship, my residency training in internal medicine. And I decided internal medicine is so deeply immersed in psychiatry that one should do some training in psychiatry in order to become a good internist. So I did a rotation in psychiatry. And the thing that impressed me most was how sick depressed patients were and how much better they got when they were adequately treated with antidepressants. And that actually excited me more than treating people with renal failure or a variety of other medical conditions. And so at the end of my medical residency, I switched to psychiatry and have spent the rest of my time working in this field. And one of the consequences of depression is that in the United States, there are over 40,000 deaths per year due to suicide, and at least half of those people died in the throes of depression. So that's kind of the motivation. Now, what do we know about the etiology of mood disorders? First of all, the bedrock of our confidence in understanding the etiology of mood disorders lies in the fact that there's a significant genetic component. And the second element that has emerged over many years is that we see that adverse childhood experiences are linked to greater risk of depression in adolescence and adulthood. So these two twin factors operate and account for the depression in most people. Now, how do we figure out what is wrong with our car engine, right? The first thing you do is you lift the hood and the diagnostician examines the engine directly. And now, in fact, they just take their computer, their laptop computer, and plug it into a USB port in the engine to try and figure out what's wrong. And this is the kind of thing that medicine does in general in order to understand what is the cause of a condition. And our tools in depression and psychiatry in general are to do autopsy studies of the brain, examine the brain directly, and do brain imaging studies of people while they're alive. These are the two tools that we have for looking under the hood to find out what's going wrong. And I'm going to try and give you some illustrations of how fruitful that approach can be. So in the 1970s, psychiatrists began to come up with ideas about what might be the etiology of depression. And there were the theories of a deficiency of serotonin or norepinephrine, the so-called indolamine and catecholamine hypotheses. And so this had great currency. It was a kind of a deficiency, biochemical deficiency in the brain, which gave the treatment of mood disorders and the concept of mood disorders something that sounded medical and worked really well in conversations with patients, reporters, and so on and so forth. But the truth is that we actually understood very little about what was going on. Other elements were introduced progressively over the years and in parallel to understand the pathophysiology of mood disorders or depression, the HPA axis, overactivity, glutamate excess, GABA deficiency, and deficiencies in neurogenesis and excesses in inflammation. So all of these parallel courses have been pursued in research. Oh, that's not too good. Back. Yes, good. So we started to look at the fundamental hypothesis of serotonin deficiency because SSRIs became the predominant antidepressant treatment that was being used worldwide. And they were kind of predicated on the idea that they're correcting a deficiency in serotonin. So we decided to try and follow the path of neurologists who looked at, for example, dopamine neurons in Parkinson's disease and found a distinct deficiency. And amazingly enough, there hadn't really been quantitative morphometric studies of serotonin neurons in the brain of depressed patients. So we carried out these studies. When I say we, I mean particularly my colleague Victoria Rango, who's a neuroanatomist, did this kind of work. And what we found was, as you compare the depressed suicide brain on the right and the control brain on the left, we found the exact opposite of what our hypothesis was. We found more neurons, those big black blobs. They're darker, as you can see, and the processes in those neurons are also darker. So this amount, and the stain that we were using for this was for tryptophan hydroxylase, which is the rate-limiting enzyme for the production of serotonin, the neurotransmitter. And so we found more neurons, more tryptophan hydroxylase per neuron, and more serotonin, which I don't show you here, in the neurons. This was the exact opposite of what had been predicted by the indolamine hypothesis of depression. We also did a similar study with Victoria Rango and Mark Underwood, counting noradrenergic neurons. If you have noradrenergic deficit, you would say maybe there's something wrong with those neurons, not enough. And so we used that same kind of method, and we quantified all the noradrenergic neurons in the locus, in the brainstem of patients. And we found that there was a deficiency this time, actually fewer neurons. But only this axis along the x-axis is like the brain stem, and the neurons at the upper end of the brain stem project to the brain, and the neurons in the lower part of the brain stem project to the spinal cord, and the deficiency was only in the neurons that project to the brain. So that was really fascinating, and that actually supported the noradrenergic hypothesis. And you can see how you can take the guesswork and the speculation out of all this by looking under the hood and counting what counts, the neurons. Next. So that raised a number of things. We became interested in what about brain growth, what regulates the production of these neurons, and so on and so forth. Now, one of the very interesting things that one learns is that the brain uses different things for multiple purposes. So, for example, the serotonin system. Serotonin is a neurotransmitter, but it is also part of the trophic processes in the brain, working through various trophic factors, growth factors, including BDNF, brain-derived neurotrophic factor. So serotonin plays two roles in the brain. It is involved in the growth of the brain and the multiplication of cells, and it is a neurotransmitter involved in mood regulation, cognition, appetite, sleep, etc., etc., etc. Wow. Okay. So a previous prize winner for BBRF was Renee Henn. And Renee Henn and Alex Dranovsky published this very nice paper review in Biological Psychiatry in 2006. And what you see here is the dentate gyrus in the brain of mice. Now, the dentate gyrus is one of the few places where you have what's called adult neurogenesis, where the brain makes new neurons. Now, in mice, it only makes neurons through middle age, but in humans, it goes on making very efficiently new neurons into the 80s. And so when you stress mice, here is the dentate gyrus. It's called the dentate gyrus because it looks like a tooth. When you stress them, you can see fewer neurons, right? There's a loss of neurons. And the processes, which include synapses at the end of these things, all over them, the processes shrink. It's like hair loss. So you have fewer neurons and you have hair loss. And then if you give antidepressants, you can block this. Actually, have a look at this closely. You not only block, you overshoot in terms of neurons, actually extra ones. And you can see that the processes extend and become like this. So this was kind of their model. Neurogenesis is the way antidepressants work. Okay, they may do that through serotonin, they may do that through a variety of other pathways, but the common final pathway is neuronal growth. Now, the other interesting thing you see from this is that, well, this is the depression story using this model that they were using in mice, and you can see it's the opposite effect. So you're kind of correcting an abnormality, but nobody really knew if this was an abnormality that existed in depressed patients, and nobody really knew whether antidepressants had the capacity to do this in depressed patients to reverse these morphological effects. And so we decided to check this out, and we did this. My colleague Mara Baldrini did all the work. I come up with ideas and other people try and make stuff happen. So this is what we did. We have a brain collection, a bit like you heard from Dr. Schwartz in Maryland. We have a similar brain collection. And here is we first looked at the volume or the size of the dentate gyrus. This is what it looks like in the human brain. And here is a green, this is the volume, the size of the dentate gyrus in controls. This is all post-mortem. And look at the volume in depressed patients, half the size. And look at this. This is depressed patients who are on SSRIs at the time of death. They look like the controls, a little bit higher, but basically the same as controls. So you have half the volume when you're depressed, and you have a normal volume when you're on SSRIs, and this is exactly what you would have predicted from Renee Hen's mice. What about mature granule neurons, which are the major neuronal type inside the dentate gyrus? You find that here are the controls. There's the number of neurons in the controls. These are the depressed patients. They have a little over half the number of neurons, but you can see far fewer. And here are the depressed patients on SSRIs. These ones are on TCAs, tricyclic antidepressants that hardly anybody uses anymore. But you can see a bit of normalization. But the SSRIs seem to be able to reverse this whole thing. And then, here's an interesting study. We looked at the progenitor cell count. These are the cells that go on to produce new neurons in the dentate gyrus. And here are the, most of this effect, by the way, just like in the mice, is in the anterior part of the dentate gyrus, not in the posterior part. So the mice and the man, if you like, human look like, behave similarly in terms of this anatomical specificity. But here's the healthy controls. Here are the depressed patients with this significant reduction in the number of multiplying progenitor cells, which suggests that the problem is at the fundamental production of neurons in depressed people. And look what happens when you give an SSRI. You've got this huge increase. So this is how depressed patients catch up. When they get treated, they catch up by overproducing the progenitor cells, which then accelerate the production of mature neurons, and this is how they catch up. So when you treat depressed patients, or when the depressed patient is sitting there depressed, these key parts of their brain are shrinking, they're losing neurons, and when they are treated with antidepressants, this process begins to reverse and continues while they are taking those medications. By the way, for people who love to take benzodiazepines along with their antidepressants, that is a really good way, this is what the black is, that is a really good way of retarding the action of the antidepressants. The patient may feel calmer, but their brain is getting better at a much reduced rate. Okay, now remember the processes, you know, they shrink and then they expand. When they shrink under depression, they expand under antidepressants. Mara Baldrini looked at the processes, and in fact, this is exactly what we find. We find that the depressed people have shorter processes. So when you look under the hood, you can find a great deal. Oh, that must be... Okay, now to understand what is really going on with this. So part of the reason we think this is... now you begin to see serotonin tied to neurogenesis, right? It's SSRIs who work specifically through enhancing serotonin that produce the neurogenesis and restore the dentate gyrus volume and restore the number of mature neurons, because serotonin is part of a trophic brain growth cascade. Now we need to tie this back to what happened to our famous indolamine serotonin deficiency hypothesis. We just showed you at the beginning that everything seemed to be in the other direction. We have tons of serotonin. Depressed people have more serotonin and more serotonin neurons than non-depressed people. What's going on? So we decided to look at regulation of serotonin firing and release. Now, with PET scanning, you can see the serotonin neurons right here because they are covered in a 5-HT1A receptor, which is an autoreceptor here. Over here, this receptor in all these cortical areas and subcortical areas, it's a postsynaptic receptor. It's part of serotonin signaling. It mediates, to some degree, the brain growth effect. But over here, it's an autoreceptor. This is the brainstem, and this is exactly where all the serotonin neurons are located. And as an autoreceptor, it provides feedback onto the neuron to shut off firing. When the serotonin neuron releases serotonin, it does all its signaling, but part of it goes in a little feedback loop back onto the cell itself and shuts off firing. And the reason is that the brain is very intolerant of serotonin deficiency and excess. Excess produces psychosis, hence you just heard it's not a good idea to give schizophrenics and psychotic people tryptophan because it makes them more psychotic. But in terms of mood, you don't want to have a deficiency because then you're depressed. Right? So the brain regulates the serotonin at a very narrow range to keep it at just the sweet spot, Goldilocks firing range, and it's all regulated by these, or a lot of it by these autoreceptors. So what do we find when we looked at the autoreceptors? First, in the postmortem brain, which is where we always go first, we found that in the upper part of the serotonin neuron nuclei in the dorsal raphe nucleus, there is an excess of 5-HT1A autoreceptors. Excess. And then Renee Hen had made this mouse, because, I think partly because of our findings in the humans, and this is a mouse that overexpresses the 5-HT1A autoreceptor specifically. And that overexpressing autoreceptor mouse has lower firing rates of serotonin and releases less serotonin and has a behavioral phenotype of depression. So, armed with that information, and here's the depressive phenotype, the forced swim test, the mouse swims less because it gives up faster, and the tail suspension test, it stops wiggling around and struggling faster. We looked in patients with a PET scanner, and Ramin Parsi did a lot of this PET work, and we found that depressed patients, which is shown here, not recently medicated depressed patients, have a lot more red, including here, you can see this is the autoreceptor, and that's because they have more autoreceptors. Yeah, I'll be done. Which is exactly what one would hypothesize, more autoreceptors. We found this was true in depressed patients who had MDD or bipolar disorder, and we found it was a trait because it was present during episodes and between episodes. And here's the amazing thing. Matthew Millak, a colleague of mine, looked at the offspring of our depressed patients and found that the offspring, which is shown in the hatch, looked like the depressed patients. And in fact, the offspring who went on to develop depression, when we follow them up, had the highest autoreceptor levels. So there are genetic and epigenetic reasons why this gene is overexpressed, genetic and epigenetic, so we can understand how genes and environment both can produce this phenotype. And treatment. What do SSRIs do? The SSRI's common element of effect is to downregulate this autoreceptor. So that this is not only the pathology of illness, but it is also the main target of SSRIs. Now, there are many models of depression that have evolved over the years, but they're all highly linked. I've already mentioned the serotonin trophic effect, but we see other types of growth factors come into play. We've heard about GABA deficiency, glutamate excess, and ketamine works as a trophic factor. It promotes the growth effects of glutamate, blocks its toxic effects. It has its antidepressant effect in hours or days, but it works through an enhancement of processes and an enhancement of synapses. So you can see it all funnels down to the same thing, the trophic effects. And these are all correlated with each other. The serotonin abnormality is correlated with HPA axis overactivity and correlated with the glutamate stuff. And in fact, we can even image inflammation, which may damage the brain. And here is there's a link between depression and inflammation. And you can see here, scanning, this you can scan effects of inflammation in the brain. And the greater the severity of the depression, the greater the severity of the inflammation. And we've heard from the previous speakers how that translates itself into neurotransmitter effects and potential brain damage. And so our treatments can target these very abnormalities. So I'd like to just say brain imaging is powerful. It bridges research between mice models that are crucial and the human brain. You can actually see what is wrong. You can understand how antidepressants correct these abnormalities. And you can develop better treatments that can do the same thing faster and more reliably, like ketamine. And here are my colleagues that did this work. Thank you.
Thank you. So, John, I have a bunch of questions, but I think we've unfortunately run out of time. Maybe I and other people could buttonhole you at the end of the conference to ask them, okay? Because it was really a fascinating presentation and great interest. So